Hi, my name is Jason Yang, and I'm an assistant professor at Rutgers, New Jersey Medical School. I am a faculty member in our Department of Microbiology, Biochemistry Molecular Genetics, and in the Center for Emerging and Reemerging Pathogens. Today, in this lecture, what we're going to be talking about is the problem of antimicrobial resistance and how some of our recent advances in machine learning can help us, number one, better understand the mechanisms that underlie resistance. Number two, develop approaches for better diagnosing resistance. Number three, developing approaches or guidelines for better managing resistance in the clinic. And finally, number four, developing new drugs and therapeutics for treating resistance infections. And what I'm going to start with is talking about the global problem of antimicrobial resistance. Antimicrobials really form the cornerstone of modern medicine. And there's a pressing and urgent need to address resistance because the development of resistance outpaces our ability to develop new antimicrobial drugs. In fact, there was a recent study published in The Lancet back in 2019 that estimates that roughly 5 million of global deaths in the world are associated with antimicrobial resistance, and roughly 1.2 million can be directly attributed to resistant infections. So this is a really important problem. And in fact, what you can see in this, in this map here is that for many of our really important infections, such as Klebsiella pneumoniae, resistant forms of these pathogens have overtaken infection in many continents, including Africa and Asia. Moreover, there are a number of pathogens which are recognized by both the CDC and the WHO as urgent threats to human health. And each one of these pathogens have been identified as urgent threats because they exhibit different forms of antimicrobial resistance. And so these are, in general, referred to the escape pathogens, which include Enterococcus, Cyanobacter, Staphylococcus, Enterobacter, and Pseudomonas. But in addition to these, another pathogen which often gets overlooked is tuberculosis, and there are increasing and concerning rates of multi-drug resistant, extensively drug resistant, pan-drug resistant TB. Now, how do antimicrobials work? For most antimicrobials, these are small molecules that target processes and machinery that are involved in bacterial cell replication. So these may be things such as fluoroquinolones, which target some of the machinery that's involved in DNA replication, or protein translation inhibitors that inhibit ribosomes, molecules like beta-lactams that interfere with processes related to bacterial cell wall synthesis. And in recent years, other compounds that inhibit processes related to bacterial energetics, including molecules like bedaquiline, which target ATP synthase. So within this wide diversity of antimicrobial compounds, we now think of these as broadly two classes. One on the left are antimicrobial compounds, which we refer to as bacteriostatic. And the reason we do this, we make this reference, is because when bacterial cells are treated with these antimicrobials, the overall effect of these is that the cells cease to grow. So they experience growth inhibition. In contrast, on the right are antimicrobials that are identified as bactericidal. And they're defined this way because upon treatment, these antimicrobials can actually cause bacteria to die, oftentimes by multiple orders of magnitude, instead of just cessation of growth. And some of the reasons why these are important is because number one, in the context of pathogens which may occupy site of infection, bacteriostatic antimicrobials will really just maintain the size of that bacterial population until the immune system can come and clear those pathogen cells, whereas bactericidal antimicrobials will actively kill and decrease the abundance of pathogens that may be at a site of infection. Now, when we think about antimicrobial resistance, broadly speaking, most of the mechanisms that I've attributed to resistance really deal with processes or mutations that interfere with the ability of a antimicrobial drug to really effectively bind its target. And so there are a number of ways in which this can occur. So for instance, in the top left and top middle, there are different sort of mechanisms that are related to drug transport, whether it's difficulty in having antimicrobial get into a cell or processes that actively pump those antimicrobials out. In the bottom, there are processes related to mutations either on a target or near a target, which effectively prevent the antimicrobial drug for binding, from actively binding and inhibiting that target. And there can be other mechanisms like you see on the top right, which include overexpressing a target so that the effect of binding some of those target proteins is really blunted. So this is a way in which antimicrobial resistance is canonically defined. But there exists other modes of phenotypic resistance, which are very much relevant to the problem of antimicrobial resistant infections in the clinic. And so, some of those versions are depicted here. When we think about resistance in a way that's classically defined in the previous slide, what we're really talking about is genetic mutations in, again, transport mechanisms or genes related to or adjacent to drug targets. And the net consequence of these for the pathogen is that what we can observe by measurements of drug susceptibility, here on the top left, denoted by MIC, which is the minimum inhibitory concentration, or if you think about the context of, like, say, a dose response experiment, that first concentration that prevents growth of those bacteria. That what you can see is that for classically defined resistant cells, these cells exhibit a very large increase in this concentration. So in other words, it takes a very large dose of the antimicrobial to elicit a either growth inhibitory or lethal phenotype in those bacterial cells. But in the middle on the right-hand side, what's depicted here are other forms of phenotypic antimicrobial resistance, whereby either mutations or changes in gene expression, or even in the physiological energetic state of a cell, of a bacterial pathogen, these cells exhibit no change in their MIC or in their drug susceptibility, but demonstrate decreased efficacy in this case, in clearing the pathogen cells when using a bactericidal antimicrobial. And so an example in the middle are tolerant cells, which are still susceptible to the effects of bactericidal antimicrobials, but which die in this blue line at a much slower rate than normal drug susceptible cells in the green. And on the right-hand side are what we refer to as persistent cells. And the concept around persistent cells is that in an infection, in the context of a bacterial community, there may be multiple subpopulations of cells, some of which are actually recalcitrant, which are actually insensitive to the effects of the antimicrobial, and therefore, after, say, a certain amount of time of treatment, there exists a significant fraction of the cells that are able to survive. And so one of the active questions in research concerning antimicrobial resistance is really understanding mechanisms that are related to these types of phenotypes, as well as strategies that we can use to overcome them. And one of the most exciting developments in biomedical research has been, these really compelling advancements in machine learning and artificial intelligence that have really manifested over the past decade. And to very quickly summarize what machine learning can do. So machine learning is, can be thought of as the practice of developing predictive models that can take, different kinds of data inputs, whether those are numbers, sequences, or even information that you might have from, say, electronic health records, to build these predictive models that can predict some set of outcome phenotypes. So those could actually be, those can again become those can be quantitative measurements, but they can also be qualitative traits such as, whether or not, for instance, whether or not an antimicrobial, whether or not a pathogen is susceptible or resistant, or whether or not a patient is going to do well or do poorly in response to certain kinds of antimicrobials. And when we think about, different sort of activities that machine learning algorithms can do today, broadly speaking, these can be defined within 3 different categories. One on the left is supervised learning, whereby the data that's used, that's given to train a model, has information on both, the component measurements, which in the machine learning community are called features, as well as the predicted output traits or phenotypes, which in this case are called labels. And some of the ways in which supervised learning is useful is that we can use these types of activities for performing tasks like classification. Again, for instance, is this pathogen drug susceptible or drug resistant, or regression. For instance, what might be the quantitative response to different doses, or over time, with respect to antimicrobial treatment. A second major class is our algorithms that perform what we call unsupervised learning, where we can train a machine learning model, but we don't have phenotypic outcomes, or in this case, where we don't have labels. And where these approaches are very useful is if you have very, very large training data, for instance, very large epidemiological or very large experimental data sets of many observations. And what we can do is we can perform clustering analyses to see how many different subgroups there might be in this larger data set. And finally, there are advances that are taking place in reinforcement learning, which really develop innovative advances in neural networks to train machine learning models with both data that has been measured, as well as other data, some of which might can actually be generated by the machine learning model itself. And so, what I'm going to do now is switch gears and talk about some of the ways in which machine learning has really, which has already been applied towards addressing the important problem of antimicrobial resistance, and some of the opportunities for machine learning to continue into the future. And so the first of these is in understanding the mechanisms that are related to resistance. And as I described a few slides ago, right, resistance can be defined as both, conventionally defined mutations that interfere with the drug-target interaction, as well as, say, physiological changes in the cell that can confer tolerance or persistence. And with respect to how the microbiology community, understands or thinks about antimicrobial action, depicted here, there's a growing appreciation that in addition to the primary effects of an antimicrobial binding its target, in this linear chain on the left, whereby target inhibition can lead to disruption of essential cell processes that initiate a cascade that directly leads to cell death, that there are also physiological consequences of binding a target, for instance, increased metabolic activity, that can elicit secondary modes of phenotypes such as bacterial cell death, which may occur through events such as the accumulation of toxic reactive species. And some of the most exciting recent research has been understanding how some of the physiological effects of binding a antimicrobial target can lead to increases in metabolic demand that really initiate these kinds of cascades. And so one of the open challenges in this field is really understanding what might be some of the specific mechanisms that drive some of these features here, such as increased metabolic demand and increased metabolism. And there have been some really exciting recent publications that have proposed ways in which machine learning analyses, such as supervised machine learning analyses, such as regression and classification, can really be used to help us understand some of the pathways that underlie these processes. And what these involve, in some of these newer approaches, what they involve are the usage of semi-comprehensive or semi-comprehensive network models of bacterial physiology, such as metabolism, to interpret data that you might get either from, in the top, activities such as biochemical screens, or on the bottom, data from activities such as whole genome sequences of pathogen strains, to make predictions on the top specific metabolic pathways that are involved in the efficacy of different antimicrobials, or on the bottom, different genetic biomarkers that can predict which strains might be resistant or susceptible. And one of the reasons why this is really exciting is because identification of these metabolic pathways and genes can potentially lead to discovery of new drug targets for future antimicrobial drug discovery. Now, a second topic that I want to talk about is how machine learning activities can really support our ability to diagnose antimicrobial resistance in the clinic. And so currently, the ways in which these are being performed is through processes which we refer to as antimicrobial susceptibility testing. And basically, the way which antimicrobial susceptibility testing is conducted is to take pathogen cells cultured on either a broth media or an agar plates that you get from directly from an infected patient, whether that's by collecting a sample from the site of infection or by collecting bacteria, for instance, that may be coughed onto some of these materials, and then testing for, on the left-hand side, testing for growth of the antimicrobials in the presence of different, testing for growth of those bacteria in the presence of different antimicrobial agents using a battery of different kinds of assays, which on its own is very slow. Or number two, in the middle, performing assays that amplify and sequence for genes that are already known to be associated with resistance, thereby looking for known mutations, which again can be very slow over the matter of days. Or finally, other kinds of experiments, such as by mass spectrometry, that can look for protein or metabolite or other kinds of biomarkers that can uniquely identify resistant strains. Now, some of the ways in which machine learning is facilitating and really advancing and accelerating diagnostics is in really accelerating the ways in which we can identify biomarkers for resistance that go beyond identifying either specific known mutations or by just phenotyping a small number of curated resistant strains. And so what's depicted in this figure are in the top, microbes that you can collect either from patients or here that you can collect from space and in some of our, here, I guess, in the International Space Station. And several, and this idea that we can actually, for these microbial strains, say it's susceptible resistant strains, we can collect many different kinds of data, which may be, complementary or orthogonal to each other, and then train deep learning, machine learning models to really get high fidelity predictive models that may not necessarily depend on our knowledge of our comprehensive knowledge of resistance conferring mutations, and that these activities can be sufficient for predicting resistant phenotypes given a number of these complementary mutations in the clinic. Other ways in which machine learning is advancing our ability to diagnose resistance are other approaches that can help us deal with noisy data that we can get from some of these different experimental approaches, and again, new algorithms that can predict human phenotypes, human outcomes directly from some of these measurements, rather than just making predictions on just the pathogen cells themselves. So moving forward to clinical management of resistant infections, one of the ways, I think one of the most important features or considerations of dealing with antimicrobial resistant infections is that resistant infections can very quickly lead to the development of sepsis and death, severe morbidity or mortality of the patient. And some of the obstacles to diagnosing and treating resistant infections rapidly in the clinic is that most clinicians make clinical decisions by following patient responses to a number of clinical interventions. And so one of the ways that you might think about this is, for instance, like a clinical flow chart for clinicians to make decisions. And during each of those phases between decisions, can take multiple days. So, for instance, for treating infection where it's unknown whether or not the infection is susceptible or resistant, a clinician may first treat a patient with antimicrobials that the infection is actually resistant against, and wait to see if there's a response. And in fact, like and even diagnosing which pathogen the patient may be infected with, they may even cycle through multiple antimicrobials, and you can imagine how just on that on its own can lead to poor patient outcomes. And so having to go through this process of making decision series can severely limit the ability of clinicians to diagnose and quickly act on resistant infections. And moreover, some of the misuse or overuse of antimicrobials can also lead to many other undesired effects in the human patient, such as collateral diseases related to antimicrobial disruption, what are otherwise say health promoting or good populations of bacteria that help support human health. And so I think a very prevalent example of this is in how antimicrobials can really disrupt the bacteria that live in the human gut and support many aspects of human health, including nutrition. And what's been shown extensively is that antimicrobial misuse or overuse can disrupt the microbiome and lead to diseases such as inflammatory bowel disease. But there's several other examples of this. And so one of the ways in which, one exciting ways in which antimicrobial machine learning is facilitating clinical decision-making and antimicrobial stewardship is to develop and apply these new, oftentimes neural network-based approaches to analyze, the very large volume of electronic health records for different patients which include information about, say, symptoms of different kinds of infections, as well as oftentimes information about different measurements that be made in patients, such as heart rate variability, temperature, lethargy, other kind of symptoms, and to use these types of information to make patient-specific diagnoses and predictions for treatment strategies that can lead to favorable outcomes. Now, in this last bit, what I want to do is talk about how machine learning has been, accelerating, facilitating and accelerating the ways in which we are now discovering new therapeutics that optimistically can address this growing problem, antimicrobial resistance. And first, to give some context, I think it's important to understand that although antimicrobials have been around for roughly 100 years and have been used in the clinic for roughly 100 years, most of the antimicrobials that are used in the clinic today were discovered, more than 30 years ago. And one of the major challenges facing antimicrobial drug development is that over the past 30, 40 years, we are in this period which has been described as this antimicrobial discovery void, where many of the newly discovered compounds either fail in efficacy over previous compounds, or for which resistance in pathogens develops so quickly that these antimicrobials are no longer thought to be good solutions for treating different kinds of infections. And so there are a number of ways in which machine learning is now being applied to facilitate antimicrobial discovery. One of these on the left are approaches that algorithm approaches that directly build predictive models of, say, small molecules to learn features of these small molecules, which can be used to these chemical features that can be used to predict chemical features of candidate antimicrobial agents, whether those small molecules currently exist or currently already FDA approved for treating other sort of diseases, or for assembling new molecules based on some of these antimicrobial characteristic chemical structures. A second area in which machine learning is being applied, in the center column, is toward this important topic of drug-target interactions and better understanding why certain mutations may elicit antimicrobial resistance, and better understand from a biophysical, biochemical perspective, what might be good positions in, say, a drug target that might either enhance the performance of existing antimicrobials, or that may serve as good novel binding sites for future drug targets. And I should say, also related to some of the some on one of the slides earlier about discovering antimicrobial resistant mechanisms, that also, again, those efforts, machine learning guided efforts, can lead to discovery of new drug targets themselves. And finally, on the right-hand side, are activities in which machine learning has really been guiding our ability to develop new therapeutic approaches and non-small molecule based therapies for treating resistant infections. And these include things such as antimicrobial peptides, for which machine learning activities are now being developed to help predict protein structures for putative or candidate antimicrobial peptides. Machine learning has also been recently developed and applied to facilitate the discovery of naturally occurring antimicrobials. So, for instance, from either the human microbiome or other sort of bacterial communities such as soil microbiomes. And the ways in which these activities are being conducted is to take whole genome or metagenomic data from these different bacterial communities in the search for biosynthetic gene clusters, which underlie the ability of bacteria and other microbiological organisms to create previously uncharacterized molecules, which can then be tested in the laboratory to evaluate if such undiscovered small molecules may have antimicrobial properties or activity. And so some of the reasons why this is actually a very exciting approach is that many of the antimicrobials that were discovered about a century ago and are still actively used today, such as penicillin, right, streptomycin, were discovered by observing or by kind of serendipitously stumbling upon kind of antagonistic interactions between different types of microbial organisms, right, whereby one organism creates one of these inhibitory molecules to suppress the growth of other bacteria growing in their environment. So this is very exciting because this is a chemical space that is incredibly unexplored, and there's a lot of enthusiasm about how these approaches can lead to discovery of completely brand new classes of antimicrobials that have never been explored before. And finally, there are many exciting therapies that are now being proposed to address this problem of antimicrobial resistance, including not only the discovering of new drugs or antimicrobial peptides that directly target the pathogen, but also many other technologies such as the use of bacteriophages or drugs that target the patient or the human host to, for instance, to potentiate the immune system or to really inhibit some of the symptomatic features of infection that lead to high morbidity or high mortality. And machine learning activities are being applied in each of these different, exciting therapeutic areas to really design those kinds of molecules or protein or genetic interventions, which largely as a community, there's a lot of enthusiasm that this whole consortium of approaches can really help us address these critical problems facing our future. And so with that, I just want to summarize that what we've talked about today is this really important problem of antimicrobial resistance, and how machine learning is helping us address problems related to understanding mechanisms underlying resistance, problems related to diagnosing resistance in the clinic, problems relating to making decisions about how to treat resistant infections, and finally, problems related to how we can discover new therapeutics for treating resistant infections. And with that, I want to thank you for your attention.